So we've developed a bone health domain. What we're going to go through today is just an introduction to, again, review why bone health is important. Just a reminder of, osteoblast activity, osteoclast activity. We have an actual orthopedic surgeon on this call, so I won't take too long for fear of boring him. And then we're going to talk about the 7 aspects of bone health that the Aristotle monitors, the metabolites underneath those 7 aspects, and how it's going to be used to infer specific factors underlying bone health. Then we'll go over some clinical applications and how it can be used. We'll go over a case study, not our own, since we do not have any instances of this test being calculated in individuals, but we do have some simulated data. And we'll show that for the 21 metabolites that we propose to use as markers of bone health, 3 of them already have significant associations with fracture risk when we do fracture risk modeling, predictive modeling with the data we've already collected. And then we'll talk about what the report will look like. We have a mock draft of it right now for that page, and it still has to go out to developers so that we can automate it and create the full report. So, of course, why does it matter? Well, we have 200 million people worldwide that suffer from osteoporosis. Global prevalence is 19.7% with a very tight confidence interval, 95% confidence interval. We know that roughly 33% of women and about 20% of men over 50 will experience some sort of osteoporotic fractures. In Europe, they spend 37 billion euros per year. This was, I believe, back in 2020. 2020, they spend about 37 billion euros per year on healthcare costs just related to osteoporosis. And the US spends a disproportionate $19 billion annually for healthcare costs related to osteoporosis. And on the right, you will see this graph. On top, you'll see the prevalence of osteoporosis. And on the bottom, on panel B, you will see deaths associated to fractures. So quite a bit. The companies, the countries that do the worst in this space are India, United States, Japan, Germany. And so some countries do a lot better although they have a high prevalence, they have a relatively low death due to fractures. Countries like Canada, of course, are doing a lot better, or Brazil. So why are we doing this? Right. So DEXA, dual energy X-ray absorptiometry, it can detect bone mineral density loss before it can be identified on usual skeletal radiography. So it's possible to diagnose osteoporosis in postmenopausal women and older men before clinical features arise. However, there's limitations. The main thing is that this is a static assessment, right? So DEXA scanners are generating a 2D image of really complex 3D structures. And they report bone density as a quotient of the bone mineral content divided by the bone area, right? So an obvious pitfall of this method is that the larger a bone is, it will convey superior strength, but may in fact have the same bone density as a smaller bone. It also has very limited predictive power. So although osteoporosis in adults is diagnosed based on a T-score equal to or below minus 2.5 standard deviations, most individuals who sustain fragility fractures specifically are above this often arbitrary cutoff. And so there's an incongruity here that poses a challenge to you clinicians in identifying patients who may benefit from osteoporosis treatment. And there's also a lack of personalization, as we know. So what we know is that while DEXA is the gold standard test for measuring bone density, and guys, we're measuring DEXA in our XPRIZE participants. This isn't going away anytime soon, but clinical judgment should take precedence if results sort of contradict clinical information, right? So we have a few things that we can look at, for instance, with metabolites. We can look at procollagen type 1 N-terminal propeptide, P1N. This is going to be reflecting the synthesis of type 1 collagen, of course, the main component of bone matrix. We can look at bone-specific alkaline phosphatases. These are sort of the other traditional biomarkers out there, right? It's an enzyme that's associated with osteoblast activity and bone mineralization. We can look at osteocalcin, which is a protein that's secreted by osteoblasts, the bone-forming cells, during bone formation. It also influences energy metabolism. And so there are obvious detriments to even the traditional biomarkers we use when we look at C-terminal telepeptide. It's a marker of, again, type 1 collagen degradation. When we look at N-terminal telepeptide, when we look at tartrate-resistant acid phosphatase 5B or TRAP5B, this is an enzyme released by active osteoclasts, and so it's linked to active bone breakdown. We can look at calcium and phosphate regulators, right? Serum calcium, phosphate, vitamin D metabolites, specifically 25-OH vitamin D, and even parathyroid hormone. So you as physicians obviously know the influence of parathyroid hormone on vitamin D and calcium cycling, the activities of those hydrolase enzymes in the liver and the kidney. We won't get into that right now. But these are the traditional biomarkers. As you can see on this comparative chart we have here, they're limited. They're not going to detect early changes in bone metabolism. They're not going to have lots of insight into bone turnover. They're not going to be able to tailor to an individual's overall metabolic profile. And oftentimes they don't reflect the real-time metabolic status of that bone. So one thing that we know, we're going to have some take-home messages sprinkled out throughout today, is that current diagnostic tools like DEXA, they provide limited insights, and there is a need for precision approaches, right? So here's a quick review of bone biology, right? So when we have this process of synthesis and the mineralization of the bone matrix, osteoblasts are responsible for this, and both during initial bone formation and then later during bone remodeling. We have some key molecules here to discuss, right? We have type 1 collagen and osteocalcin. So osteoblasts produce type 1 collagen. This constitutes the majority of the bone matrix, as I mentioned earlier, and osteocalcin, the non-collagenous protein that plays a role, again, in bone mineralization. The support of hydroxyapatite deposition. So the matrix produced by osteoblasts becomes mineralized with hydroxyapatite, a crystalline structure composed of calcium and phosphate, and it provides bone with strength and rigidity. And then we have the breakdown of bone matrix to release calcium and phosphate. So osteoclasts are these really large multinucleated cells. They resorb bone by secreting acids and enzymes, and this often leads to the release of stored calcium and phosphate directly into the bloodstream. That's why we test for that, right? And then we have tartrate-resistant acid phosphatase, TRAP, and cathepsin K. So these osteoclasts will secrete TRAP and cathepsin K. These are both enzymes, again, that are essential for degrading the organic components of the bone matrix. And then there's a balance in bone remodeling to maintain a structural integrity, right? The activity of these osteoclasts is usually balanced by the activity of the osteoblasts to ensure proper bone remodeling and maintaining bone strength and structural integrity. And there's this dynamic balance that is really dependent on this bone homeostasis and coordinated activity, as we can see here. And really homeostasis of bone is achieved through the coordinated actions of both of these cell types and ensuring both continuous remodeling and adaptation to mechanical stress. And also we should take note of the imbalances. So there's disruptions that lead to osteoporosis or other bone disorders, and it's a result of this imbalance in the activity of these cells, usually characterized by a decrease in bone mass and increased fracture risk. Did I skip one? Okay. So here are some key pathways related to UNT signaling. And on the right, you'll see a KEGG diagram with those key pathways labeled. These are all within the 68% of metabolism that the Aristotle test can have insight into. So looking at things like protein digestion and absorption, citrate cycle, looking at glutathione metabolism, arachidonic acid metabolism, these are all mapped. Obviously, you can see the TCA cycle. But we're also looking at other really important pathways like WNT signaling, Rankle and rank signaling pathways, TGF-beta signaling pathways, MAPK signaling pathways, P13-ACT signaling pathways, and calcium signaling pathways. And then, of course, probably one of the most important is osteoclast differentiation signaling pathways. And so those are other KEGG identifiers that have been mapped onto this figure you see on the right. And this is where most of our discussion today, and this is the metabolic circuitry that most of our discussion today will be focused on. So here we see metabolites in bone health. We know we'll talk about what these all are, like hydroxyproline, citric acid, glutathione. These are the really big ones. So we have 21 metabolites that will be in this panel. And 3 of them, again, when we simulate data with our own collected data that we have up to this point, we see that 3 of them already have statistically significant associations in the direction we would expect given previous research in the field. And those 3 are these, that we're going to cover right now: hydroxyproline, citric acid, and glutathione. And down here, we see a relevance score. This is a score that has been developed recently in precision medicine. Recently, I mean 2-3 years. And this score will essentially represent the importance or significance in bone health based essentially on their roles in different processes. So when you see hydroxyproline, we know that its importance, its relevance score is about 80% in collagen turnover. Citric acid has a relevance score about 70% in mineralization. The antioxidant glutathione has about a 60% relevance score in antioxidant defense. And linoleic acid has a 50% relevance score when we talk about systemic inflammation modulation, which is a big deal related to bone health. So this is, again, a relevance score that has been sort of mined from the data on the available data that we've collected today, and we'll discuss in short order. And it's basically it's essentially a mixture of the significance of that metabolite between experimental groups, as well as the variance accounted for in that metabolite. So hydroxyproline, when you see that, you should think collagen turnover. When you see citric acid, you should think mineralization, glutathiones related to antioxidant defense, and linoleic acid, really systemic inflammation modulation. So what do we have here? DEXA, again, it measures bone mineral density. It's at a single point in time, provides really limited insights into dynamic processes like bone turnover. It does not account for systemic factors such as inflammation or oxidative stress. And there's a poor ability to predict fracture risk in patients that have normal BMD values. Now, on the flip side, metabolomics, it's going to capture real-time data on bone remodeling processes, including formation by looking at osteoblast activity, a resorption by looking at osteoclast activity. It also reflects systemic influences on bone health, such as nutritional status. Citric acid or calcium metabolism, for instance, is factored into this scoring that we have. Oxidative stress by looking at glutathione levels, or inflammatory markers, for instance, linoleic acid. And it really enables early detection of metabolic imbalances that really precede structural bone loss, and it provides personalized insights into how do we guide targeted therapies. And so here we have an insight score, and this is another score, again, in precision medicine that's basically used to judge between available tests and imaging techniques on the market. And it's designed to allow for a gross measurement of how these are going to be used by physicians. So we have 4 different domains that are used for this. Dynamism, which is weighted 40% when calculating an insight score. Coverage, weighted 30%. Predictive value, 20%. And then you have the personalization, which is a 10% weight. And you can tell that this is definitely an index that is used to judge the relevance and benefit of various personalized or precision medicine tests, because we want it to be dynamic. We want it to have excellent coverage. We want it to have high predictive value, and we want it obviously to be personalized. So when you do something such as a DEXA, you're getting, again, BMD 1 point in time. Metabolomics data is dynamic, so it tracks ongoing biological processes, including bone formation and resorption markers over time. So we are going to give DEXA basically a 30% here because it's a static and it's focused on BMD. And then we'll estimate metabolomics here at 90% because, again, we're looking at collagen turnover, oxidative stress, et cetera. For the systemic coverage, we know that DEXA is limited to local bone density. It ignores systemic factors like inflammation, oxidative stress, or nutrient deficiencies, whereas metabolomics can account for these holistic factors that we know play an important role on bone health. DEXA will give it a 20%, ignore systemic processes. Metabolomics will weight at 80% as it captures systemic influences. And then, again, we're doing this same thing with predictive value. Again, DEXA is retrospective. It's not going to predict future bone health or fracture risk without significant pre-existing bone loss. And metabolomics, whereas metabolomics is going to really predict metabolic imbalances or risks even before structural damage occurs. So if we weight DEXA 40% here, fracture risk is basically estimated on thresholds like the T-score, whereas the 85% that we've given metabolomics here is capable of detecting early metabolic dysfunction. And then again, DEXA offers little to no personalization as far as treatment planning, because all you're looking at is bone mineral density, and you're really ignoring the 6 other factors that we think play a role in bone health. So we'll give it a 20% there because of general thresholds for all patients. And overall, I think we can say conservatively that metabolomics probably is going to give about 50% more insights than a traditional DEXA scan. So yeah, early detection is going to be key here. Our goal is to identify subtle metabolic changes in collagen turnover and bone mineralization. Our goal is to detect bone health deterioration before structural damage or fractures occur. And really our aim is to enable the proactive intervention by you, the physicians, to prevent bone-related conditions like osteoporosis. So for instance, elevated hydroxyproline levels indicate increased bone resorption. And then we also want you guys to track treatment efficiency, so you can monitor real-time changes in metabolic markers during the therapy. You can help evaluate the effectiveness of treatments like bisphosphonates, vitamin D, calcium supplements, and also it provides feedback to adjust the therapeutic strategies for optimal outcomes via digital twinning. And again, we can integrate systemic factors, so the score can be interpreted in the context of other domains. Again, linking bone health with these influences like oxidative stress from an inflammatory domain, nutritional status, looking at citric acid for mineralization. This offers a holistic view of the patient's metabolic health. And then with our digital twinning, we can provide these really tailored insights into not only their metabolic profiles, but giving lifestyle recommendations and targeted therapies to optimize their bone health outcomes. So the take-home message is, again, Metabolomics is going to provide a dynamic system view of bone health, surpassing traditional static biomarkers. So we have 21 metabolites that we've indexed for this domain, and they are across these 11 chemical subclasses you see in the pie chart. But why these markers, right? So markers like citric acid, oxoglutaric acid, collagen-related amino acids like hydroxyproline, these are directly related to osteoblast activity and bone mineralization. And then also markers like hydroxyproline and taurine, they provide insights into bone degradation and oxidative stress, which accelerates resorption. So looking at osteoclast activity. And then, of course, we have metabolic support. So even Krebs cycle intermediates like succinic acid, they reflect energy production that are actually crucial for bone forming cells. So there are even metabolites of the TCA cycle that indicate osteocyte activity. And then we have inflammation and modulation of that more specifically. So lipid metabolites, things like linoleic acid, oleic acid, as well as their counterparts, antioxidants like glutathione. These are going to help us assess systemic influences on this bone turnover. So here we see sort of the domain of bone collagen turnover. We know that collagen is going to make up the organic matrix of bone. And amino acid levels, certain amino acid levels can reflect collagen turnover. We know that a key marker of hydroxyproline, the key marker of collagen degradation, I should say, is hydroxyproline. And we know that elevated levels usually indicate bone resorption and increased turnover. And we'll see in the simulated data that we've run on the data we've collected so far, when we do fracture risk predictive modeling with levels of hydroxyproline, we do see that same relationship. So with increased hydroxyproline, we have increased fracture risk. Next metabolite in this domain is going to be lysine. It's going to play a role in collagen cross-linking, critical for bone strength. Arginine supports nitric oxide production. It's also going to play an important role in osteoblast activity. And cysteine, they contribute to antioxidant defense, right? So this is really a key factor in preventing osteoporosis. And on the right-hand side, we do see the production of AGEs, right? These advanced glycation end products. These are formed in vivo via the so-called Maillard reaction. So reactive aldose or ketose sugars and metabolic intermediates like 3-deoxyglucosone, glyoxyl, or methylglyoxyl. These will react with free amino groups in lysine, hydroxylysine, and arginine, and they lead to the formation of a Schiff base, which undergoes rearrangement to form a relatively stable Amadori product, as you see here. So this is the activity specifically of arginine in this cross-linking for bone strength. Yeah, this next domain we see here is really interesting. So we know citrate or citric acid, it's essential for bone mineralization. It actually will stabilize hydroxyapatite crystals. Oxoglutaric acid, this is obviously alpha KG, 2-oxoglutaric acid, oxoglutarate, whatever you want to call it. I think it has like 19 synonyms, but it is going to support not only cellular energy production through the TCA cycle, but it's required for osteoblast function. And in fact, we can see on the protein structure on the right, when we look at the crystallography data for, Robert, do you have a question? Maybe it was a false raised hand.
He's muted. I apologize, I was muted. One quick question. Would you consider glycine synthesis as the part of a collagen? It seemed that we have, at least what I read, that glycine seemed to be pretty key factor, especially synthesis, because as far as I understand, I'm not a metabolomics expert, but it's not regulated by its demand, but a lot of people have a really low glycine, and that has a lot of consequences. Some people would say that glycine is conditionally conditionally essential amino acid because we have limited ability to synthesize, and most people don't eat enough glycine, hydrolyzed collagen. Do you think that we could add this? This is just because I agree everything what you said in terms of metabolism of the bone, but I understand bone is a collagen that is also just like starch. It's a collagen that has been hardened by hydroxyapatite. So it may give us some insight if you don't have a collagen problem, like glycine can be, methyl transfer, there is good from the different aspects. Would you think that would be, I can give you some articles if you want to, on importance of glycine in metabolism, bone, and collagen.
Absolutely. So here's the thing. We looked at glycine, and you're absolutely right that glycine is a hugely important molecule, not just for the generation of collagen, but also in terms of glutathione metabolism, which is in the panel. So glycine is a very important molecule. And in some regards, it will be calculated for like the nutritional influence on bone health.
And
Keep in mind,
cardiovascular with a heart and collagen and fragility of the vessels and stuff. So I don't want to steal your thunder, but I'm just asking if you guys would consider
we
if
absolutely can. We just need the research for it. So when I went out to look for metabolomic specific data that's looking at glycine and some aspect of bone health, I did not see very high effect sizes. So although it is implicated absolutely in both collagen synthesis as well as glutathione recycling, I cannot find data that shows really high effect sizes when you look at either cell studies, animal studies, human studies of glycine. And that was one of the molecules that there was a few others that we kind of investigated, but because of a lack of very high significant effect sizes with large magnitude, we abandoned it as far as trying to look for it. So if you have some data though,
I will provide some articles that was just
absolutely.
time. I don't know look at metabolomics, but I look at it because I suggest to my patient take collagen and my like especially athlete to recover soft tissues. So we would recommend take 10 to 15 grams of hydrolyzed collagen 1 hour before practice. And the whole process is to subsidize glycine, optimize glycine nutrition. So I have tons of articles in that respect. I will share with the group. I don't want
Please.
to.
Yeah, and this isn't going to launch for the next, few weeks. So we have time to tinker around with models and feed new algorithms into our AI. And like I said, right now we're looking at these 7 domains that we'll come back to again. But like right now, this is bone mineralization. Glycine may play a role. I may have overlooked something. We may have failed to find a study that can link that with strong associations to bone health. But if there is one out there, we absolutely want to include it. And then we want to obviously check for model fit because we don't want to assess something with 7 different parameters. The likelihood of that being validated and generalized to new testing instances is very low. So we always want to be thrifty in our modeling and use as few parameters as possible. So going back to oxyglutaric acid, this is a metabolite of the TCA cycle. And as you can see, it actually fits right above the T loop there. There's the folding pocket. So when we create this protein structure from its crystallography data, we do see that alpha Kg is required. And it only binds if the pocket for magnesium ATP is filled in already. So it is dependent on both magnesium and ATP levels. And then we have malic acids, indicative of cellular metabolism. It's again linked to the bone forming cells, osteoblasts. Okay. And this kind of invitation to submit data, this goes to everybody. So if you have studies on certain metabolites that we monitor, like glycine or serine, and we can include that in our model and show its relevance and predictive accuracy to different aspects of bone health, please submit that. We are in the business of synthesizing literature to leverage insights. So this would be excellent. All right, so bone remodeling, right? We have lipid metabolism, which is basically going to cause a lot of systemic inflammation. Linoleic acid and alpha-linolenic acid, these are both precursors of prostaglandins, and they will modulate bone remodeling inflammation. You have markers like stearic acid and palmitic acid. These levels will indirectly reflect dietary influences. And then oleic acid, this is a mono unsaturated fatty acid, and it's protective of bone. It's actually implicated with reduced inflammation. So dietary, here what we see on the right-hand graph is basically dietary and endogenous palmitic acid metabolism. So we have dietary, let's go through this figure. Dietary palmitate is, again, first emulsified, then hydrolyzed in the duodenum. It becomes a free fatty acid, or it can become a monoacylglycerol. That's then going to be absorbed by the enterocytes, the free fatty acids, and mono-AG. These are, again, reesterified into triglycerides in the cytosol of the cell. They're incorporated into chylomicrons, transported into the lymph duct for transport. And then you have lipoprotein lipase. This is an extrahepatic tissue. This is going to hydrolyze that chylomicron. It will release then the free fatty acids and triglycerides, and it will produce a chylomicron remnant. I hate lipid metabolism. I hate reviewing it. When an individual is in the fed state, so when we have increased glucose, this will lead to insulin secretion that will stimulate GLUT4 and CD36 translocation up to the plasma membrane of the cell. That'll facilitate uptake of the glucose and the palmitate into the adipocyte. Glucose is going to then be, of course, metabolized to the pyruvate. That'll enter the TCA cycle. It'll produce citrate and subsequently acetyl-CoA. We have de novo lipogenesis. That'll utilize the acetyl-CoA there to synthesize palmitic acid. And that's going to be essentially the primary product of this process. And palmitic acid will be used as a precursor for fatty acid esters of these hydroxy fatty acids. They will also be used for ceramide synthesis. They'll have downstream effects, as you can go to this paper here, and this will be the citation number 2, published in the Journal of Clinical Lipidology. You can go to this and see what you can go to this paper and see the downstream effects it has. But basically, this is looking at both the average American intake, a low carb intake, and high carb intake. This is one dietary preference that will significantly modulate inflammation that will lead to enhanced bone resorption. Okay, what do we have here? Spermidine and spermine. These have been shown directly to stimulate the proliferation of osteoblasts. Again, the cells that are responsible for bone formation. So this effect contributes essentially to increase bone matrix production and overall bone health. And then in the context of bone matrix formation, these polyamines facilitate the synthesis of collagen and other components of the bone matrix. It really does lead to improved strength and density. On the right here, we see that spermidine and spermine inhibit osteoclastic differentiation, and they do not affect cell survival. So here we have pre-osteoclastic raw 2647 cells. These were cultured with rankle, either in the presence or absence of spermidine and spermine. So A is spermidine, B is spermine, and C is putrescine, actually. And these concentrations, as you can see, range from 1 nanomolar to 1 millimolar. And they're followed by the determination of trap-positive MNCs and MTT reduction. So we see the quantitative data on the left. We see some typical microphotographs on the right. And in D, we see that these raw 2647 cells that were cultured with rankle in either the presence or absence of spermine and spermidine, followed by real-time PCR, looking for the determination of the mRNA expression of these osteoclastic marker genes. And then in E, we see the primary osteoclasts, again, were cultured in rankle with or without 1 millimolar spermidine and spermine, again, followed by this trap-positive MNC and MTT reduction. So this shows that they're significantly different from each control value obtained in the absence of the polyamines added. So pretty conclusive cell studies. Again, we're going to go back to niacinamide and the B vitamins. These are going to act as these really important cofactors, vitamins and cofactors, and they support NAD-dependent processes, mostly in osteoblast energy metabolism. And then pyridoxine is particularly important for collagen synthesis, as we cannot, is a critical cofactor for collagen synthesis. We cannot create it with low B6. So in the figure on the right, we see that nicotinamide stimulated osteoblast differentiation. In the figure A, we see that ALP and alizarin red S, ARS. This is a special type of staining. It was performed in these MC3T3 cells. These were cultured in osteogenic medium, supplemented again with the indicated concentrations of nicotinamide for 5 and 12 days. The osteogenic medium containing the nicotinamide was replaced every other day. In B and C, we can see the quantification of each staining that was performed with image J. D and J will show the mRNA levels of osteoblast differentiation marker genes. These were determined again by real-time qPCR on day 10. K shows the scheme of nicotinamide treatment for RNA-seq analysis. These MC3T3 cells, again, they were cultured with 10 micromolars of nicotinamide in osteogenic medium. And the osteogenic medium containing nicotinamide was, again, replaced every 2 days until the cells were harvested. The green arrows you'll see, these indicate the days of the nicotinamide treatment. In L and M, we see correlation analysis was conducted with the nicotinamide increased DEGs on days 4 and 10. We see also a normalized correlation matrix to show that the correlation among the GO terms, so when they retrieved the GO terms in the biological process category, they had a significant correlation. The GO analysis for the gene clusters on day 4 and 10, you can see them here on the bottom line. This shows that the top 5 GOs were selected based on the adjusted p-values and then sorted by the enrichment scores. And then finally in N, we see the heat map of the genes included in cluster 4 on days 4 and 10. And all of these, most of these are significant at the p equals 0.05 level. Some are significant at 0.1 and 0.01, as well as 0.001, with very little non-significant changes. All right. Calcium and phosphate regulators. So we do not have direct measurement of calcium and phosphate, right? We are a metabolomics platform currently, and these are too small. These are elemental indicators, but we do have metabolites that reflect phosphate cycling and calcium metabolism, and they act as strong proxies. 2 of these are going to be fructose 6-phosphate and glucose 6-phosphate. Again, these are indicators of overall energy availability. and they will determine a bone cell activity. And then also we have succinic acid. These is a metabolite, of course, of TCA, and it reflects the metabolic status of osteocytes. On the figure to the right, we see those three metabolites represented. Specifically, this is looking at the metabolic fates of glucose in mammalian cells. So the major biochemical pathways you can see are denoted in red. The main product for each pathway is shown in blue. Glucose is used to produce not only energy, but also intermediate metabolites for biosynthesis. And there are several key enzymes that are highlighted in green. Please note here that the citrate can be exported from mitochondria and converted to acetyl-CoA in the nucleus. And that's going to exert some epigenetic regulation on gene expression. And so the metabolic pathways are common among different cell types, and their relative importance does likely vary on the biological function of each cell. But as you can see, these are calcium and phosphate regulators. Looking at oxidative stress, this is going to contribute obviously to bone loss, and we want to counteract this with antioxidants. Glutathione, potent antioxidant, has also been implicated in bone studies to mitigate damage, oxidative damage to bone cells. And then taurine, as we can see on the graph to the right, it has really potent anti-inflammatory effects, and in the context of bone loss has also been shown to protect against demineralization. So here we see this is just a representative scheme of the action of taurine, specifically in skeletal muscle fiber. And it's shown here that the tau T carrier, which works to maintain a very high intracellular taurine level, along with the actions that taurine exerts on membrane channels itself and the sarcoplasmic reticulum and the mitochondria, and even possibly gene expression. But there is putative binding sites for taurine. They're shown one on the SYK1 channel, and then the second place it's shown as a local anesthetic drug binding site. These arrows indicate a general stimulating action. The dotted lines are going to stand for inhibitory effects or yet undefined pathways. And then the pathway for taurine efflux under stress conditions, and these are things like ischemia, osmotic stress. These are likely being regulated by the volume-sensitive organic anion channel, VSOC, and that's also shown here. So what can we do with the Aristotle? We can use metabolomics to induce a paradigm shift for the proactive rather than reactive treatment and personalization of bone care. So who is this good for? This is going to include postmenopausal women, elderly men, individuals with chronic conditions, and it will enable you to detect early metabolic changes that are predictive of fractures. In addition to that, it's a metabolomics test, right? So we can track responses to these various treatments, whatever they may be, and provide some dynamic insights to optimize your care plans. So we have developed also, to go along with this, a comprehensive index of 100 bone health interventions. These are going to be across the 7 critical domains, and that'll form the backbone of our digital twinning approach to these problems. And these interventions are the ones we may recommend to patients depending on their individual metabolic profiles and bone health status. The 7 domains we'll look at are going to be nutrition. So examples here, we have 20 domains in nutrition, 20 in physical activity, 15 for environmental health, lifestyle, and medical prescription drugs that we may recommend. And then there are environmental and emerging therapies and research. So in nutrition, this can be anything from calcium and vitamin D supplementation, optimizing protein intake, incorporating magnesium-rich foods. Under the physical activity recommendations, which we have 20 of them, this will cover specific weight-bearing exercises, resistance training, balance-focused practices like yoga or Tai Chi. A lot of the recommendations that you're already getting. Hormonal health, menopause management strategies, optimizing testosterone in men. Of course, addressing thyroid and parathyroid hormone imbalances, as this is going to significantly impact bone health. And then we have lifestyle interventions. These will focus mostly on bone depleting habits like smoking cessation, stress management, improved sleep hygiene, reducing alcohol consumption. And then we also have some medical and pharmacologic options that we may recommend, things like bisphosphonates, denosumab, teriparatide, some other treatments for fractures and secondary osteoporosis. And then we can also give some environmental recommendations. I always like these. They're like supporting environments, fall-free, basically fall-proofing homes for the very elderly, even advocating for like community walking spaces. And then there are some really cutting-edge emerging therapies we might recommend. Not many, but these are going to be things like stem cell therapy, gene editing, AI-powered fracture risk models. And through the digital twinning, the interventions are going to be, just as before, mapped to each individual's metabolic data. This will enable, again, precise, dynamic, and personalized care plans. And the idea here is to allow us to simulate outcomes, refine recommendations for each patient's unique needs, and then help you guys transform bone health management into a more proactive science. So again, our goal here is to empower you physicians to use metabolomics for the precise diagnostics, treatments, plans, and again, proactive management. So we don't currently have our own case studies, right? We don't. We haven't even tested anybody with this domain, but there is a noteworthy example from Metabolon that does a lot of research grade testing in this space as well. And that demonstrates the potential of metabolomics in advancing, again, bone health diagnostics. So really, Metabolon conducted a research study, and it compared the metabolomic profiles of elderly women with severe osteoporosis and elderly women with high bone mineral density, right? And their goal was to identify biomarkers indicative of bone health and fragility. They found some key things. They discovered this isovalyryl carnitine, the C5, carbon 5 as a metabolite. It correlated with total tibia volumetric BMD, and it's suggestive of its role as a potential biomarker for detecting bone loss. And this is a really good use case for metabolomics. This early detection is enabled by biomarkers like this C5 that can help identify individuals at risk of osteoporosis well before the structural damage begins to occur. And then insights from metabolomics, of course, will allow tailored strategies to prevent this bone deterioration and subsequent fractures. So they didn't do that intervention part, but they did run this study with a group of physicians providing their research testing there. Metabolon is the first company to offer commercial metabolomics, and they don't do direct to consumer, even physician sales, but they do a lot of research, and they're basically a research for hire body. So Metabolon's case study really does exemplify that metabolomics can have a transformative role and play a major influence in bone health diagnostics. And so it really should inspire the field as a large to explore similar approaches for own clinical applications and really, again, aiming for personalized and preventive care. So on this slide, we see a compelling case for using metabolomics to assess bone health and fracture risk. This data is simulated based on patterns observed in our own records and was generated by integrating these known metabolite functions with fracture risk modeling. Specifically, we used metabolites like hydroxyproline, citric acid, glutathione, and modeled their relationships with fracture risk using correlation and regression techniques. And so the simulated data really does mimic real-world trends seen in the clinical studies we just presented on. So it provides a really strong framework for our testing going forward. So let's start with the heat map on the left. This data shows the correlations between key metabolites and fracture risk. Notably, anything with a bold text highlights a statistically significant relationship. Again, this should emphasize, so it's not due to type 1 error, right? It emphasizes the reliability in clinical applications. For instance, hydroxyproline, which is a marker of collagen turnover, has a strong positive correlation with fracture risk. Elevated levels of this metabolite, which indicate increased bone resorption, are predictive of higher fracture risk. Conversely, we have citric acid. We know it stabilizes the hydroxyapatite. It's crucial for bone mineralization, and it shows a strong and negative significant correlation with fracture risk. So lower levels are linked to reduced structural integrity of the bone. And on the right, we see scatter plots that visually represent these relationships. So the red trend line illustrates how changes in the metabolite levels, like a drop in glutathione and citric acid, can predict fracture risk. And these patterns were modeled to, again, simulate real-world conditions and provide actionable insights into the dynamic bone remodeling processes. This is far beyond what traditional static tools like DEXA can offer. And again, this is from simulated data of our own collection. These are people who we've tested on ourselves. And it really shows the power of metabolomics and early detection. And if we can identify patients at risk of fracture before these fractures occur, you physicians can intervene proactively with targeted therapies, whether it's dietary recommendations, supplements, medication. So in conclusion, I won't belabor the point, metabolomics isn't just for this advanced diagnostic tool. It is a way to personalize and elevate patient care. It is an endeavor to move us closer to precision medicine, and here specifically in the bone health management. So again, evidence underscores the utility of metabolomics in transforming healthcare. So what is this going to look like? Well, right now we've collaborated with you all to provide some updates into our testing. This was recommended by Gail and Robert. You guys requested this and we went and found it. Robert, we will discuss the inclusion of glycine and any other markers that, again, add significant predictive value without necessarily, and we haven't really shown the models. There are statistical models behind these, but we want to increase the predictive value without increasing model fit. the model to not be overfit. So every single metabolite you use, they're like statistical bucks, right? If you use more metabolites, you're going to get more accuracy, but that model will necessarily be less generalizable and relevant to new testing instances. So if you can be thrifty in your model development and still have high predictive accuracy, it's always advisable. We are developing this training module for you guys so that you can use this metabolomic data effectively. We will start to pilot it with you and gather your feedback. And then again, we have the 100 interventions that we want to include as part of our digital twinning for this health domain. And we still have to go through and spend lots of time indexing things like, effect size, directionality of change, 95% confidence intervals, effect homogeneity, looking at beta coefficients, baseline changes, everything that is needing to be modeled in that input into manually curated into that training file that we then feed to our neural engine. That file has to be updated. That will take some time. And then the long-term vision, again, is to predict fractures, assess bone health dynamically, guide these personalized treatments that you guys will implement with your patients, and hopefully see enhanced patient outcomes. As I mentioned, we are seeing with retesters, given 85% compliance, we are seeing significant improvements in overall score and every single domain except the gut and mutational load. The mutational load is improving, though not statistically significant, and the gut health, unfortunately, is deteriorating at retest, although not statistically significant. So we want to keep gathering data on this, and with retesting, we should see if our efforts are paying off. And then continue building the database again for implementing new models, whether it's glycine or implementing new therapies and treatments that you may have in your clinic and you want sort of twinned on your patient's metabolic profile. This is what the report page will look like. So as you know, we have gone away from the purple enrichment plots. These are now showing numerical number lines that will show the low reference average and high reference average, as well as that patient's levels. And then we have talked about these 7 domains that underscore bone health, and this is how it will be represented: bone collagen turnover, bone mineralization, bone remodeling and inflammation, bone growth and repair, bone density and resorption, calcium and phosphate regulators, and oxidative stress and bone health. Every single these are models, right? These are 7 models that we've developed, and every single model has a 90% predictive accuracy on the mined and simulated data. When we do internal validation through permutation testing, specifically Monte Carlo leave one out cross validation, we see the metabolites, the models are not overfit. So these models are going to most likely be generalizable to new populations that they're not trained on, i.e. your patients. And we do see that they have high predictive accuracy when we, again, take these levels and we corroborate them with known outcomes from the literature. And this format for reporting the domain, this will be something that we continue with all of our domains. All of our domains will now start building out the factors underneath them. So for aging, you'll see things like senescence, autophagy. In cardiovascular domain, you'll see things like ion traffic channeling. You'll see things like thrombosis, endothelial function. In the mitochondria, you'll see things like fission fusion. This was already there. These were already a part of the domains, but we are building it out so you can have greater interpretation rather than trying to extrapolate it from what the digital twinning recommendations are making. Now, bear in mind that we will have sort of a moratorium for a couple of weeks to decide what to do with this, collect more data from you guys, and any recommendations. If you have a specific type of therapy, we'll share with you the 100 therapies we have indexed. And if you have a specific therapy you do not have in this list, let us know. In the next few weeks, we will index it, we will model it, we will collect the data for it so that we can simulate it on your patient's profiles and produce that as a recommendation if it's fitting. And we still have to look over glycine, right? So this is still in tandem, but given us a couple weeks, this will start being reported in your own customers' data. And so you have joined the frontier. Hopefully, you'll adopt metabolomics for bone health. Again, we're moving away from gut health. Not that we're not predicting things like alpha diversity and gut barrier integrity and the gut axes, the 3 gut axes we monitor, but we are not moving the needle at retest. And we kind of figured we wouldn't when we set out to do this. Now we have a really sufficient pool of retesters right now, and we're looking over their data. And XPRIZE kind of was the impetus to make us look at the data. We have not had time to look at our own retester data, but looking at that has really shed light into the fact that we're not moving the needle on the gut health. You do have a request for bone health. we think we can make lots of impacts in this space. And it's timely because we are launching a gut health test soon, right? January 15th, it will go live. And so it is probably best that we stop looking tangentially into the gut using the blood metabolome. Although there are great papers, I can share with you papers that have looked at alpha diversity with high predictive accuracy in the blood metabolome, published in Nature Biotechnology. It's a fantastic paper, but that's not the issue. It's not that we're not assessing gut health. It's that we feel we're not improving it. So we're going to move forward with bone health. And then we have, a contact list. If you guys have any questions, you all have my text, my number. Feel free to text me, call me. You can also get a formal Zoom meeting by just going to therio.me/questions. But in the next couple of weeks, like I said, we will start moving away from gut health and start implementing this bone health domain, which we think has really high predictive accuracy and it's pretty thrifty model development.
Well, because the bar is so low, you're right. We look at DEXA and that's it.
Yeah.
And we don't know anything about what's going under the engine. It's like looking at my picture and says, yeah, you're gonna die.
Yeah, exactly. So I definitely want to iterate with you, Robert, concerning glycine. Glycine and a few other metabolites, like I mentioned, were on our list. But when we looked for effect sizes, they didn't have as much predictive accuracy and they added one less degree of freedom in the model, which means that it's going to be prone to overfitting. So we can reassess it if you have studies that I was not able to crawl.
I just don't know. My limited knowledge is that glycine is kind of a fingers in a lot of things. Glutathione,
Absolutely.
metabolism. That's a rate limiting factor. And I personally, I don't know whether Gail recommend the patient the collagen or not, but I gave people collagen, and my semi-scientific or pseudoscientific explanation is that we are doing it for glycine because every third amino acid in collagen, which is the number one protein, is the glycine, and it has a lot of stuff, and we cannot synthesize it enough. And a lot of people say like, well, yeah, you can make 3 grams of glycine, you can synthesize it, but we need about 15 grams optimally if you exercise and stuff like that. And now we see new research in fascia, which is now like a new musculoskeletal tissue recovered. Now everybody has a fascial stuff. So I think that I will look into it. I don't know answer. Maybe I'm sure that you're right. And I don't want to overdo it. I would just bring it to attention that, and you guys look at it. But I think that bone health, it's tremendous. It's a half of the population. Women has tremendous amount of problems. 2 out of 3 after 60 and 1 out of 3 after 50 has osteoporosis. And so if we can get better, even better than DEXA, which might not be like an airplane, where just it will be significant for a lot of patients.